Hello, dear students. I hope you're fine and doing well. Here we have to talk again geochemical cycling, especially nitrogen cycle, uh, the basic structure and basic cycle change we have studied in previous class. If you remember, like here, nitrogen is present in ammonia form or it is in any other intermediate form, nitrate or nitrite. So these are converting it one form to another form. And we know, especially this thing very well, that nitrogen is the major molecule of the living, major element of the living organisms, or living molecules or biomolecules in form of amino acids. Nitrogen, which is present in environment, can be converted into NH3. This NH3 can be converted in NO2 and NO3, and the process is called nitrification. So this NH3 can also be assimilated and used by the living organisms and for the production of the amino acids or proteins in living organisms. It also can be used in form of NH3 and also can be used in form of NO2. This process is called assimilation. Because when nitrogen is taken from environment for the growth of plant and animal is called assimilation or fixation. And in living organisms, it is present in form of amino acid and proteins. When any living organism dies due to decomposition, this ammonia, this nitrogen releases to the environment to form NH3 and process is called ammonification. This is aerobic process and here it is anaerobic process. Similarly, NH3 in anaerobically also absorbed are taken up by living organisms for the product for the formation of proteins in form of amino acids. And the process similarly will call assimilation in which ammonia is converted into amino acid or NH2 group of the amino acids and the decomposition of proteins and the formation of ammonia is called ammonification or nitrogen fixation. This nitrate again converts into nitrite, converts into elemental nitrogen and from elemental to NH3 and this process is called denitrification. It is one is directly and in another it is indirectly. So we know very well that amino acids or nitrogen is present in amino acids in form of amino proteins from dead cells and waste products. Okay. When any living organism dies that contains proteins, and the proteins are made up of amino acid, and amino acid contains a huge amount of NH2 molecule. So what happens? When any living organism dies and going into the soil that is decomposed by microorganisms and the proteins are separated and degraded into amino acids. This is going into environment. Okay. Almost all nitrogen in the soil exists in organic molecules. There is no elemental nitrogen in soil. Primarily in protein, when an organism dies, the process of the microbial decomposition results in the hydrolytic breakdown of proteins into amino acids, this process. And these amino acids are again converting into ammonia. Process is called ammonification and this process is also carried out by soil microorganisms and soil fungi. The amino group of amino acid are removed and converted into ammonia. Process is called ammonification. Is brought about by numerous bacteria and fungi. If we are looking here, ammonia is converted by the ammonium ion NH4 is converted through soil microorganism called nitrosomonas into NO2, which is called nitrate ion. And this nitrate ion further converted by another microbial group and genera that is called Nitrobacter. Nitrate ion by Nitrobacter is converted into nitrate ion, NO3. The genera Nitrosomonas and Nitrobacter are autotrophic nitrifying bacteria. These organisms obtain energy by oxidizing ammonia or nitrate. In the first stage, Nitrosomonas oxidize ammonia to nitrite. In the second stage, such organisms as Nitrobacter oxidize nitrites to nitrate. So, if basically, if we will understand from this talk, we want to say that elements which are essential for life are present in complex form in organisms, in biomolecules, in organic materials. When is this organic material or this living organism is going to die or dying? After that, it is decomposed by microorganisms and converted into basic elements. These elements are going to their origin. Some are going to environment, some are going to soil. And from soil, these are coming into the water, are in soil solution, which is called a SAP solution, and available for new growing plants and organisms. Either those are microorganisms, either those are plants. So, elements which are converting in one form to another form. The basic responsibility of microorganisms is there to convert these complex organic molecules into their elemental form. So microorganisms are called scavengers of this globe. They have responsibility to maintain this ecological system, to clean waste material, dead material, to recycle waste material into to obtain their basic elements, to convert waste material into elemental form for reuse. In one way, we can say these are scavengers. In one way, we can say these are reusers or recyclers, which are recycling waste material to produce it is original elements in their pure form to the environment, where these essential elements will be used for the generation of the second life. Suppose key process and prokaryotes in the nitrogen cycle. Nitrification process takes place in soil. This is process and example of organism. The process of nitrification, in which ammonia converts into nitrate. First, ammonia will convert into nitrite. Microorganism is involved, nitrosomonas, and nitrite will be converted into nitrate. Microorganism is involved, called nitrobacter. So in every step, microorganisms are responsible to convert these nitrogen intermediate forms into its other form. Denitrification process, in which NO3 nitrate converts into elemental nitrogen, and Bacillus and Pseudomonas species are involved in this process to remove the oxygen from nitrogen molecule. And N2 fixation, nitrogen fixation, which is present in environment, nitrogen plus hydrogen plus, it will form ammonia and H2, NH3 plus H2. So free living organisms will work for this. Free living organisms are responsible to convert environmental nitrogen into ammonia, which is usable because direct nitrogen most of the plants cannot use. Free living organisms are working. If environment is aerobic, azotobacter and cyanobacteria will capture environmental nitrogen from environment and they will form ammonia in bacterial cells, and this process is called nitrogen fixation. Aerobically, azotobacter and cyanobacteria will take environmental elemental nitrogen into body and will convert them into NH3. If process is anaerobic, so Clostridium purple green bacteria and Rhizobium, these will take environmental nitrogen and will convert it into NH3. In symbiotic reaction, Bradyrhizobium frankia are responsible to convert this nitrogen. Ammonification, another process. First was nitrification, second was denitrification, third was nitrogen fixation, and now that in these stages nitrogen has been converted into other forms, one form to another form, one form to another form, and elemental nitrogen has been converted into organic form. And now whenever these organisms are dying, process is called ammonification, in which organic nitrogen is converted into NH4. Many organisms can do this, especially fungi, bacteria, etc. When dead material, living organisms are dying, going to soil, microbial communities are going to decompose it, and microbial communities which are present inside of the living organism, they are also degrading it and converting organic nitrogen into NH4. So second is sulfur cycle. Sulfur is also essential element of the living organism. Sulfur is present in many amino acids which are backbone of the living organism. Suppose sulfur is present whenever in living molecules, in organic molecules is called organic sulfur. After death of the organic living organism that is going to soil, process of mineralization takes place, and sulfur which is present in organic form converts in H2S. This is gas. Okay, this H2S gas is going to be converted into elemental sulfur from where hydrogen is removed, and this process is called sulfur oxidation. Different bacterial species are involved if environment is aerobic. Thiobacillus, Begotia, and uh, Thiothrex. These organisms are responsible to convert H2S into elemental sulfur. If anaerobic environment is there, chlorobium and chromatium species are responsible to convert H2S into elemental sulfur form. This elemental sulfur form again can be converted in H2S. This elemental sulfur can be converted into SO3. This is called sulfur oxidation, and this further can be converted into sulfate SO4, and SO4 can be absorbed, can be fixed into organic molecules, and that is called sulfate reduction or sulfate assimilation. If intermediate species, we will see sulfate can be converted into H2S by means of sulfovibrio, reduction, or sulfate dissimilation. SO3 also can be converted in H2S, and bacterial species are involved: Alternomonas, Clostridium, Desulfovibrio, and this organism are involved to convert SO3 in H2S. So this is the complete life cycle of sulfur in environment, in soil, in environment, in marine water, while H2S produces after the death of the animals. This H2S converts into elemental sulfur. Elemental sulfur is going to change into sulfate. Sulfate is going to be changed into uh, SO4, and SO4 is available for assimilation and fixation in living organisms. So if you will see the main key steps and the process of sulfur cycle, number one, sulfur sulfide or sulfur oxidation, in which H2S converts into elemental sulfur, and after it converts into SO4, which is soluble into water and can be absorbed by plant roots. If there is aerobic environment, these sulfur chemolithotrophs are responsible to convert this H2S into elemental sulfur and then it into sulfate. And bacterial species are nominated over here are Thiobacillus, Begota, and many others. If aerobic environment is aerobic, purple and green uh, phototrophic bacteria and some chemolithotrophic bacteria are responsible to convert H2S into elemental sulfur and then into sulfur. Sulfate reduction aerobically SO4 into H2S. Disulfovibrio and disulfobacter are responsible for this step. Sulfur reduction anaerobically from elemental sulfur to H2S. Disulfuromonas and many uh, hyperthermophilic archaea are responsible to convert elemental sulfur into H2S. Sulfur disproportionation, disproportionation means complex sulfur conversion into H2S gas as well as into sulfate. So this sulfovibrio and other species are taking a part in it. Organic sulfur compound oxidation or reduction, in which suppose this sulfur is present in organic molecule or in amino acid, it is converted into carbon dioxide plus H2S and DMSO to DMS. Desulfurylation, a removal of sulfur from organic compound and to form H2S. Many microbial populations are taking a part over here. So it was our talk. So it was our talk regarding essential elemental cycling in environment. So if you will see, if you will conclude this lecture, this geochemical cycling in which we want to say either that is in terrestrial system, either that is in aquatic system, organic molecules which are composed of many molecules, many elements. Suppose in living organism, if we will see, living organisms are made up of carbon, nitrogen, oxygen, sulfur, phosphorus. These all things are present in living organism. When this living organism is dying, that is going into the soil because there is no any environment where it will go. Every living organism must have to go into the soil, where microbial communities are responsible to convert this dead material into its elemental form. And some gaseous elements, those are going into environment, and some micro minerals such as calcium, magnesium, carbon, these are going into the soil, and these are soluble with water. These are present in water and insoluble form. These are absorbed again by primary producers such as plants. So this is going there. This is a life pattern of this globe. I hope you have to understand and under something you have got from this presentation. For detail, you have to go through book to understand this these cycles. You have to draw these cycles in your notebook and have to try to understand. If you cannot understand some steps, you can go through book. You can go some on your research article. You can go. You can discuss with your colleagues, and you can also ask to your mentor by any available resource to in this environment. Thank you. Have a nice time.